Következő előadónk dr. Andrea Ciliberto. Néhány szóban bemutatom őt, mielőtt meghallgatjuk az előadását. 2024. szeptember 1-ével kapta meg az egyetemi tanári kinevezését. Biológus, aki 2000-ben szerzett PhD fokozatot, 2022-ben pedig habilitált. 2022 óta kutatófejlesztő az információs technológiai és bionikai karunkon, előtte pedig dolgozott a Budapesti Műszaki és Gazdaságtudományi Egyetemen tudományos főmunkatársként, dolgozott Olaszországban és az USA-ban is. Az NKFIH OTKA sejtbiológiai bizottságának tagja 2023 óta. Munkája során a sejtosztódást, ezt az élet alapját képező mechanizmus tanulmányozza. Kísérleti munkát és matematikai eszközöket kombinálva azon dolgozik, hogy feltárja a tumorok kialakulását befolyásoló sejtosztódást megváltoztató mechanizmusokat. Előadását angol nyelven tartja meg, a címe Cells Machines Made to Divide, és kaptunk viszont, ha jól tudom, mindannyiunknak a rendelkezésére áll egy magyar összefoglaló az előadásnak a tartalmát illetően. Gentile professore, grazie di essere uno dei professori della nostra università. Auguri per il titolo ricevuto di professore e io la prego di fare la sua presentazione.
Nagyon szép lenne adni az előadást magyarul. Szerintem vicces lehet, de biztosan érthetetlen. Akkor azért olaszul sem jó ötlet, szerintem, mint poki kafém. Azért angolul lesz, sajnos. Szerintem a legközelebb magyarul, biztos. So I work on cancer research, and in fact I work on resistance. So in this plot I show you in qualitative terms the problem. So on the y-axis you see cancer growth, and on the x-axis you see time. And so the cancer increases in size until the treatment starts. Maybe. No. Maybe this, yes. Until the treatment starts. When the treatment starts, the cancer decreases in size. And so initially, treatment is very successful, typically. But then what happens is that the tumor increases in size again. And the reason is that there is a subpopulation of cells that is born around this time that is insensitive to the treatment, does not respond to the treatment. And so the tumor growth increases again. So this is the problem that we study. And in order to explain to you how we try to study this problem, I give you also a bit of history, but I think it's a bit unusual that an Italian professor is in Pasmanin, so I want to give you a little bit of background. So why I started to work in Hungary? I started to work in Hungary to work with Professor Novak Bela. So Bela he unexpectedly died at the end of February. He was a very well recognized and influential Hungarian scientist who was working in Oxford, and when he died. But in 2002, when I started working in Budapest, he was at the Technical University, as you heard. And so Bela was an engineer, but he was also interested in biological problems and was really a pioneer of applying mathematics to biology. And in Bela's group, we were working on cell division. So cell division is this process whereby one cell becomes two cells. So I hope all of you may remember, so this would be a cell. This is the membrane. This is the nuclear membrane. Inside you have these molecules that are called chromosomes that have the DNA, so the genetic information. And the process of cell division is really a process whereby you start from one cell and you make two cells identical to the first. And so in order to do that, the first thing you have to do is to replicate DNA. So you see that this DNA structure from one, they now have two molecules. And then the point is that you have to give the two molecules to the daughter cells. And this is done through a physical process that I show here like this. So these chromosomes are really pulled by structures, tubes, that are called microtubules that pull them towards the daughter cells. So this is what they do. And then at the end of the process, you indeed have two new cells that are identical to the original cell. Now this is a this was my drawing, but the movies are nicer. This is a human cell, as you can see by the little man there. And so in red now you have DNA, and in green you have this structure that we discussed before. And you will see something a bit similar to what I was drawing in these cells when they start dividing, like this one. And we will have more of these dividing cells. So this is a very beautiful process. It has to be super well organized, and this is really the topic of the talk and the work done in my lab and the work that was done in Bela's lab at the time. Now, cell division is really very important at every level. Here I show just a few examples. For example, this is a frog egg, already two cells. It originally was only one cell. And cell division is the basic process that it keeps dividing. You will see this huge volume in many. No. So now you see this periodic process of division, and then this is, you start from one cell, you build a whole organism. This is trivial, but still amazing, and I think very little understood by biologists. So this is a role for cell division during development. But cell division is also important in somatic cells, so in grown organisms, like all of us in this room. So this is a crypt in the colon, and you see here there are some cells. Every little structure here is one cell. Here there is one dying cell represented. Here there is one dividing cell, a bit similar to what we saw. And the key point you have to understand is that these cells die and they have to be replaced. And now by an estimate, I don't know how precise, but around that ballpark, around 300 million cells in our body die every minute, meaning you have to replace them. So in each of your body, mine as well, in this very moment, many, many cells are dividing. And so there is a role for cell division that is also important in homeostasis, in maintaining the structure. There is also a role in cell division that is not so positive, and this is cancer. So now you have two tissues. Up here you have a normal tissue, and this instead is cancer. These are tissue from human colon. Every dot here basically is the nucleus of the cell. And if the dot is brown, it means that the cell is dividing. And you see that while in normal tissues the dividing cells are quite few, in cancer tissues you have a lot of cells dividing. So this is the problem of cancer, that cell division is not anymore regulated. It's not in the interest, let's say, of the organism, but cells start dividing unregulated, and eventually they will lead to the death of the organism. And I will tell you later more about cancer. Going back to our work in Bela's group, at the time, we were not actually working neither with frog nor with humans, but we were working with a model system, which is yeast. Now, you will see yeast cells, they are less spectacular than human cells, but they also undergo a process of cell division, you see they make bud, this particular kind of cells. They are called budding yeast. So very different morphologically from human cells, but they have a lot of advantages. They're easy to culture in the lab. They are easy to modify genetically. They are cheap. To keep them is cheap. And the experiments can be very, very well controlled. Now, of course, you can do experiments in yeast, but then you hope to discover something that is relevant for humans. And these yeast, if you don't know the word of the organism, but they are really the same creatures that are used for beer, for wine, for bread, fermentation. So these are organisms that are commonly used in biotechnology. And here we are saying to use them in the context of cancer or understanding cell division for humans. How is that possible? So to understand that, we have to be aware of the fact that yeast and humans are actually much closer than what you would think. They are closer for very basic properties, like cell division. If you take some other, for example, fermentation, probably they're not so similar for metabolism. But cell division is such an essential process that is shared among eukaryotes, and yeast and humans are both eukaryotes. And this paper that was published by a Nobel laureate in the 80s, I think, basically shows this conservation very, very well. So they have yeast cells, they remove one gene essential for cell division from the yeast, and they replace it with a human gene. Okay, so the yeast without the gene cannot divide. But if you give the human gene, then this gene is recognized by yeast as its own and start dividing again. Okay, so the basic machinery for cell division is very, very, very common between yeast and humans. And so studying yeast for essential problem is a very good idea. And you see it also by, here is just a list of Nobel Prizes, Lasker Prize, that is a very important prize for biological sciences. These are given to people that have been studying yeast as a model system for basic, essential, fundamental biological problem like transcription, vesicles, or endocytosis, telomeres, and so on. Okay, so with Bela, we were using yeast in order to ask very important generic problems about the cell cycle. For example, when do cells start dividing? They must know that there is enough food around in order to start dividing, otherwise they shouldn't divide. How cells know that there is a threshold, that food is enough or is not enough? Or, for example, how to make sure that the cell division process always have one phase where they double the amount of DNA and then they give it to the sister. They don't do 2 rounds of division, for example. That would be a problem. So how to make sure that once you start the cell division process, you finish it. And also how you make sure that you alternate the right series of events. And so I'm not going into details with this, but from what I told you, there are properties like thresholds, how much food is around, oscillations, how to make sure you repeat the right series of events, or irreversibility, how you make sure that once you start, you don't go back. That are very relevant, and this is where mathematics and biology were combined together. So in those years, the group in Budapest was one of the very first groups in the world, I think, and it was super successful, influential, was so successful that it dissolved. So basically, Bela went to Oxford to become professor there. Attila Csikasztagyi, who was also in ITK, he moved to Trento and then to London, and other people in other places in Europe. As for myself, around 2005, I went back to Italy and I started working in Milan. And in Milan, I work still now in this place which is called IFOM, is the Institute of the Italian Association for Cancer Research. So it's an institute devoted to study cancer. And so I told you bits and pieces during the talk. So I told you that the problem is growth. And I told you also that in cancer cells, there are a lot of cells dividing. Okay, so it's not surprising that a strategy that is usually used to treat cancer is to stop cell division. You want to stop these crazy cells that decided to divide without any control and to make them stop. The one way to do it is, for example, to alter those green structures that I showed you that pull the chromosomes apart. And so this is what you can do, and this is the use of these drugs. So this is in the case of humans. You remember the very nice movie I showed you at the beginning. You see that these cells have much less structured green structure. They don't have them. They enter into cell division and they stop, which is good. This is what you would like. These are really cancer cells. You would like these cells to die, but some die, not all of them. For example, this cell cannot really make cell division, but also doesn't, so it does something weird, strange, that you shouldn't do, because it means that he was able to do something regardless of the presence of the drug. This is yeast instead. Yeast cells also are quite different from the movie I showed you before. Again, these are yeast cells with a drug that says stop. This is a drug that is used to treat patients. They stop, but not really. I mean, they manage somehow to divide regardless of the presence of the drug. And this implies, if I go back, so we are saying that you treat them with a drug, you stop cell division, growth decreases, but there are some cells that manage to divide. And these are probably cells that eventually could become resistant cells. Okay, so the point now you understand the title of the talk, Machines Made to Divide. The goal here is to stop cells from dividing, but cells were really built to divide. Dividing is their goal. And so to stop cells from dividing is extremely complicated. And so we were thinking it would be a very good idea if this process of emergence of a resistant cell could be replicated in the laboratory. Now, if you could study live how cells at a certain point become resistant. And this is what we have been trying to do. So what I tell you now is really the kind of work that the lab, both in Milan and in Budapest, is working on. So we use yeast, and I told you why. I mean, it's very convenient on the one hand, and is probably relevant for humans on the other hand. You saw from the movie that they managed to divide a bit, not very much. And so what we do in the laboratory is we let them grow and then give them more food. Grow and give them more food. And this experiment lasts for 3 months. So there is somebody in the lab that keeps feeding these cells. What we see is really the emergence of resistant cells in time. So here, growth rate would be how fast cells divide. And here is generation, so time. This is what is called the control. So it's a cell that is not treated with a drug. They were growing at this rate at the beginning of the experiment, and they keep it at the end. They are not really affected. These instead are cells with the drug. At the beginning, they don't grow very well, and then their growth starts increasing until it reaches the level of the control. Okay, so now this is very relevant for us. We managed to observe the emergence of resistant cells in the laboratory. And then you can do many nice things. You can take cells at this time point, at this time point, at this time point, at this one, and study them, and try to understand why they become resistant. I give you just an idea for you to think what could be a mechanism of resistance. For example, you have a mutation in DNA, and this mutation is such that the drug is continuously pumped out of the cell. No? So the drug cannot work anymore. Another mechanism of resistance, it alters the site where the drug binds. So the drug cannot bind anymore and is not effective anymore. Okay, so these are, for example, two very simple mechanisms by which cells become resistant to the drug. So what we can do, we can analyze cells and ask one question that you understand is very, very important. So is there a typical way for these cells to become resistant? So is it a process that is predictable? Because if it is, that's a very good news. It means that you can think of strategies that prevent the emergence of resistance. So if every cell does something different, every time different, of course, there will not be possible to prevent or delay the emergence of resistance. But if cells instead always do the same thing, then it's a good news. So this is the question that we asked. This, I think, is the last piece of data, if you bear with me. So every column is a different experiment. And in these different experiments, so a different population of cells, we look at mutations that give resistance. And we find that in every population, always the same genes are mutated. Okay, so we do many experiments, and every time cells do the same thing. And in particular, they mutate this particular gene, which is called tubulin. So every row is a mutation. And so what we find here is that these tubulins are always the target of the resistance. There are 3 tubulins in yeast, and we find always the same genes that are mutated. Then the mutation is different, but always the same gene. Okay, so this is a good starting point. It means that you can think of ways to prevent or delay the emergence of resistance. Of course, you have to ask whether the mutation that we observe in yeast have anything to do with humans. Otherwise, this would be really something about yeast. You cure yeast, which was not the starting question. And the good news is that there is overlap. So there is overlapping between the mutations that we find and the mutations found in patients. So in red are mutations found in yeast, in yellow mutations found in patients, in gray those that are mutated in our experiment and you can find in the literature mutated in humans. These are, I mean, you cannot see anything, but these are sequences of amino acids. For example, these are mutations that are found in patients. These are mutations that are found in the experiment. But there are also, hard to see, but these are mutations found in both. And so the fraction of mutations found in both is statistically significant. So we can really use the yeast experiment to understand something about mammals. So what to do next? You can ask, as I already asked, can we prevent the emergence of resistance? Can we channel the evolution in some way? This is something that we can do and is very interesting. So we can force cells to become resistant in one way or another in the lab. Can we confirm in mammals our results? I told you yes. Can we evolve in the lab cancer cells? This is the next step we would like to do. And it would be very nice to identify vulnerabilities of cancer cells. So I told you, maybe a cancer cell becomes resistant because pumps out the drug. But maybe pumping out the drug is something that makes this cell vulnerable because that drug is also pumping out things that it shouldn't pump out. And so maybe in order to become resistant, the cell became vulnerable and different from cells that are not resistant. And so you can hope to kill specifically resistant cells. So these are all the things we would like to do. This is very complicated. Our questions need very good tools. So for example, you need, it would be very nice to follow these cells in time, to look at them at the microscope. And in order to look at them, you have to automatically segment them. You have to measure their size. You have to have tools that allow you to do this automatically because you want to do it with millions of cells. You want to be able to analyze DNA, as I showed you before, on these mutations. And you may also want to do simulations. So you want to show how cells behave in a computer and try to understand how to plan the next experiment. And so this finishes my story. No finishes, but I give you now the rationale. At this point, this is why I came back in a way. So I didn't leave the position in Milan, but I have both. And why do I have both? Because there are very important synergies, I think, between IFOM and so the Cancer Research Institute in Italy and the Faculty of Information Technology and Bionics. Because IFOM is very strong with to produce experimental data and has a strong connection with cancer. But the faculty here has a very strong expertise in data analysis, and they are very good. We are very good to produce high quality software, and there is a very strong connection with the knowledge in engineering. And indeed, so the hope is that we have the tools and the data that can be merged together, and this will give to a better formulation of experiments, understanding of this problem of resistance. And so just to tell you that these are the very last recent work. This starts to work. So we have an MSc student, Beatrix Stier, Janos, you have also in the audience, that contributed to a work where the question was, how do cells react when the drug is washed away? And we have also our Mestersteg Intelligenzia part with the software that was developed in collaboration with a group of Andras Horvat, with Gergely Szabo, on really identifying automatically these yeast cells and analyzing them. And finally, we have another very powerful thing, I think, which is the possibility of having students that belong to the PhD School of Plasmany, but they are based in Milan. And there are 3 of them, I hope soon 4. And we had several students from Budapest spending a few months in Milan, and also vice versa, students that belong to the school that came to Budapest. So this is, I think, is working and is a very nice opportunity. So I thank you all for listening. I also thank you for welcoming me in your community, which I find very nice and very stimulating. Thanks.